Ah, boom. Ah, boom. Many scientists hope that transgenic plants. Many scientists hope that transgenic plants will become an important weapon in the worldwide fight against disease. Okay. I'm trying to speak in a very like neutral, calm voice so that like I'm sure that you guys can hear me because I don't really have much hopes. Um, much hopes, much dreams, much dreams. Um, I should just download that mix honestly because I've been using it so much for studying. Why hasn't the Quran moved your heart yet? So true. So weird how that video blew up. Yeah, 151 views. Okay, blew up is a stretch, but like blew up is a stretch. Oh man, slow day so far. I mean, I'm so quiet, but it's like I don't know if that's just my speaker or if I am actually that quiet. Whatever. Who cares? I'm studying. Study means study. And if anything, I'll put a filter to increase my gain. Um, Let me just do that right now. So now, I mean, everything should be a bit louder because the gain thing is on. Maybe it sounds a bit worse, but I don't really care. What happens if I turn on my noise suppressor now? Now, do I still sound okay? I don't know. I don't really care. Okay, chill, chill, chill. So yesterday we stopped. We learned a little bit about what exactly we use those proteins we harvest from those MGOs from. GMOs, sorry. And so usually it's for drugs. These drugs can include hormones such as the growth hormone and insulin, as well as penicillin. That's an antibiotic, right? So we learned about human insulin first. We learned about how they were, obtained before, and then what were the problems with that. So usually there were two problems. One was the supply was kind of like inconsistent, not sustainable, and also that it was unsafe because well, in the case of insulin, it just wouldn't get accepted because the surface antigens would be different to the human's antigens. Um, and so it wouldn't get accepted. But for growth hormones, the problem with that was when we were using them, in order to get them, we didn't actually um, just have like, how should I say it actually? We didn't make insulin outside. We literally gave them we literally gave them. Wait, what's it? Yeah, we literally gave the children injections of the pituitary extract. And so when that pituitary extract, if it was contaminated, then that would cause a Creutzfeldt Jacob disease in the children, which was lethal, very, very lethal. So, um, yeah, that was the issue with it. So what we did afterwards was we overcame this problem by giving. Sorry, wait, what? Okay, it's a very short reason to how genetic engineering fixes this problem, but it's essentially just solving the problem of supply, where we have genetically modified organism producing the hormone in commercial amounts. That's really it. Not really much more to be said regarding how genetic engineering, what specifically genetic engineering was done. Like over here, we have an entire process of how we can genetically engineer and form the insulin molecule. Over here, it's just genetic engineering fix the problems. I mean, it's more on focusing with the problems where without genetic engineering. The reason they might be talking about this is this last point where they brought up that people bring up the concerns of using genetically modified organisms, thinking that maybe since it's on essentially natural, that it would lead to some kind of they would lead to issues. But the problem with thinking about this, or rather, that's not a problem. It's always good to evaluate whether or not GMOs are actually like safe and what side effects they might have. But it's also important to take into account the problem with not using genetically modified things. Like the alternative is to use alternate like external sources, which is like human cadavers for the extract, pituitary extract, and dead animal pancreases for the insulin hormone. And doing this was just not supply wise. It's not effective, as well as health wise. It's not always. It's not as consistent how it works. And there's a goddamn mosquito, man. Stupid mosquito. Ah. Okay. I don't know what to do. I don't know what to do. It's so annoying. Why? Why did you have to bite me right on my toe? God damn it. What blood is even present in the toe? Like, why the toe? Where is the mosquito, bro? I'm having hiccups. Oh, this day couldn't get any worse. Oh shit. Oh god damn Oh, he is back. He is back with Molam, Savlon specifically. Let's hope he doesn't, mosquito doesn't come back then. Or she, I believe, actually. Mosquitoes are so vicious, bro. Tiny little thing makes you itch so much. Should GMO mosquitoes to not bite human beings. If size is bigger than, I don't know, this yay big, do not bite. Yay big. Alright, now knowing about those two examples, let's look at a third example. Banana vaccine. So, so scientists have successfully used genetically modified organisms to make certain human proteins such as insulin and somatic equivalent, but gone through so far. However, prokaryotes do not possess the biochemistry to make more complex human proteins. Like, well, is in bacteria when we're using those to make GMOs. When we were using those to make like proteins from certain selected genes, we didn't just directly give them the DNA because they didn't have the machinery to turn that DNA into mRNA without the exons, right? The DNA which had no exons already, we call that the cDNA, and with that cDNA, they're able to make the proteins that we want, right? And so, in the same way, since these bacteria don't have the machinery for that, prokaryotes do not have the biochemistry, the machinery, quote unquote, to make some of the more complex human proteins. Now, scientists are working on ways to introduce useful human genes into eukaryotic cells, such as yeast, plants, and even animals. Yeast is eukaryotic. Okay. Oh, God. Oh, bro. Okay. Yeast, plants, and even ants. Wait, I can hold space? Oh, shit. Oh my god, that is so cool. If I press space, that brings me down. And if I press shift space, that brings me up. Okay, well that makes me so happy. Um, Many scientists hope that transgenic plants will become an important weapon in the worldwide fight against disease. What is transgenic? Transgenic. What is the circle that remains after being bitten by a mosquito? What does a larger It's just so bad. Like a bloody bro. Oh, manage the immediate skin irritation from underlying allergic response. Combination of oral medicines? Dude, lowering the symptoms is one of the fast ways to temporarily numb the area. Damp cloth and ice pack, wrapped in a towel for 5 to 10 minutes. Oh, I really want to get a ice pack. Hold on. Okay, okay, I need to stop. Avoid scratching as it can lead to super infection. Requiring antibiotics. Keep fingernails short or wear gloves at night to prevent accidental damage. Okay, okay, can we remove all mosquitoes? Like, what do they do for the, as in, like, what do they contribute to the food, food energy cycle or whatever it's called? While removing mosquitoes sounds like it might sound like a dream, they are surprisingly important causes of change. Scientists generally agree that wiping out the species that bite humans might be okay, but eliminating all 2,500 species could cause a major hiccup in the food cycle. Mosquitoes are a vital, reliable food source for a huge variety of animals, birds. Swallows and hummingbirds, bats, dragonflies, spiders rely on adult mosquitoes for energy, especially during hiding on breeding sessions. Larvae, mosquito babies live in water and are the primary supper for many fish, frogs, turtles, salamanders, and fish, including mosquito fish, specialize in eating them. Most people don't realize that nectar, not blood, is a mosquito's primary food. Male mosquitoes never like humans; they spend their entire lives visiting flowers for sugar. As they move between blossoms to transfer pollen, oh shit, they're pollinators? Oh, oh, hey bro, my bad, bro. They also act as filter feeders in their larval stage. In organic waste, algae, and tiny microorganisms in stagnant. They technically do nutrient self-forming. Oh, act as a brutal but effective natural check on animal populations. They can influence herds like caribou to migrate, preventing them from overgrazing and destroying specific patches of land. Oh, alright, hey bro, never mind. You can stay. That's funny. My back is like getting destroyed by the way I'm sitting. I God, there's so many issues. Issues. If I do. Okay, Doctor Frickin'. What is transgenic again? Transgenic organism is a living entity, plant, animal, or microorganisms whose DNA has been artificially altered by incorporating genetic material. As a specific type of GMO, a transgenic organism created using genetic engineering to introduce new desirable traits like pest resistance, future growth, or the production of therapeutic proteins. That's pretty good summary, I think. Transgenic plants will become an important weapon in the worldwide fight against disease, right? With the fact that they produce the therapeutic. It seems to be vaccination is a very effective way of eliminating serious diseases, aka like herd immunity, right? So, was it artificial or is it artificial act? No, it's artificial act. Artificial passive would be just giving them. Okay. However, less developed developed countries always import vaccines usually require storage in the fridge. In some countries, people have guaranteed access. There can also be cultural and practical difficulties to healthcare workers in allowing them to practice children. Genetically modified plants or plant products such as bananas, potatoes, or carrots carry vaccines against human diseases such as infant diarrhea or hepatitis. We can solve many of these problems. Local communities grow the relevant plants relatively cheaply, and there is no need for food storage. The benefit from healthy diseases no need for food storage. Trials are already placed in the US, USA, and China. China, bananas are emerging as a candidate for genetic modification of very fine vaccines, particularly hepatitis B. Isn't that like so cool, man? That's so. 350 million people globally are already infected with this. If genetically modified bananas could be grown carrying the hepatitis B vaccine, it would save many lives and be a great alternative to injection. Bacterium Agrobacterium syncytium is usually used to introduce genes with Bacterium Agrobacterium syncytium. I need to learn how that's called. Agro Agrobacterium tumefaciens. Yeah, it is. Human passions. So, all right. So, here are some examples of constraint plants. I mean, besides modified bananas, right? So, yeah, the bacterium, agrobacterium cultivations usually used to introduce genes from one plant into another. So this is going to be the vector, not the plasmid this time. The bacterium itself is being the vector, or is it the plasmid from the bacterium, which is the vector? Asymmetric machines causes tumors in plants, which are known as Kramgals. Contains a plasmid called the Ti plasmid, which transfers bacterial genetic information directly into the plant DNA. This is what normally causes the abnormal growth of plants, but modified plasmids can be used to carry beneficial plants into the plant genome. So the lethal bacteria, A. tumefaciens, contains the Ti plasmid, which gets incorporated into plant DNA. Now, when we modify this Ti plasmid to have the new gene to make this now non-lethal and in fact carrying the carry the desired genes for the desired proteins, then we're going to be able to carry beneficial genes into the plant genome. And then by the process of plant cloning, we can use the modified transgenic cells to produce new transgenic plants. So, G, G. Um, so the Ti plasmid is used from async machines. We have the Ti plasmid, we extract it. This is suspended chromosomes, and so over here, Ti plasmid, which has been turned to bacterium. So this is the new Ti plasmid containing the new gene. And so this is going to be non-lethal and it's going to be carrying the beneficial gene for the, you know, beneficial change in the plant that we want. So then the plant is going to be infected with modified bacterium, and then the part of the Ti plasmid will be engineered gene, which is part of the plant chromosome. Essentially, the modified plasmid gets incorporated into the DNA of the plant. And so as the cell engineered gene becomes oh, agent tumor develop in the plant. Oh my god, it hit me again. Alright, so A2 fascians usually causes a tumor to develop in these plants. These plant cells contain the new gene. If the, no, because these plant cells will contain the new gene, which is lethal. Now, if these tumor cells are taken in culture, then four new plants will be grown from them. Containing the new genes, they're genetically engineered transgenic plants. So we have something over here which is, so if you take the tumor in which caused by the new genes, take those cells and clone them to create a new plant, then we'll have a plant containing the new gene ground. So all of these cells will be containing those new genes due to aching temperature. Whereas over here, it was only this area which was done like that. Oh no. Okay, genetically modified animals. On the other hand, if you want to transfer new DNA into eukaryotic cells, but this time we want to do it as animals, it's going to be a different, more typical case. But some attempts have already been successful. Then the most exciting and recent research involves genetic engineering, has been on the work on inserting human genes into the tissues of sheep and cattle. What was the point for them? Being able to introduce genes from one type of plant into another, or from an animal into a plant. So we want to just put them into plants, right? Because at the end of the day, we want to turn a normal plant into a transgenic plant. So where these genes are coming from, um, we don't really care whether it's coming from an animal or a plant. So um, if you want to make transgenic animals, okay, so in this case, we didn't really talk about how we're modifying them or where we're getting the genes. Point is that we're going to be modifying the plasmid and placing a new gene there coming from somewhere else, which is going to be very beneficial. It's going to be coding for something else. Now, over here, we're going to be inserting human genes, right? They were talking a bit about here about inserting human genes into there. What was it? Yeah. So prokaryotes don't possess the biochemistry to make human proteins or human proteins, human hormones, human etc. But we're still working on ways to get human genes into eukaryotic cells so that they will start making you know things that we can harvest. Or another use of it would be transgenic plants, as in getting human genes into plants will allow them to be. If that is not going to be a way we do vaccination. Vaccination can be we're not putting human genes into bananas. We're putting vaccines into bananas. Like essentially the weak or inactive form of the virus into the banana, which then they eat to become, and then they'll have an immune response. So that's going to be different. Over here, however, we have some history in inserting these human genes to the tissues of sheep and cattle to produce transgenic animals. First transgenic sheep was born in 1991. An emu called Tracy, Evie, okay, called Tracy, who produced first the human protein AAT in her milk. This protein is missing in people who suffer from sheep, affecting their livers, causing this this milk develop at a very early age. So production of these proteins using transgenic animals will involve introducing a copy of the human gene which codes for that protein into the genetic material of an egg of a different animal species. So, so First, like there's the mammal. We want to make that a GMO by introducing a human gene into it. But eventually, that cell will eventually become, uh, incorporate that and start producing those proteins which would otherwise be made using a human, right? These two proteins need to be the same. Now, um, in order to introduce this copy of human genes into the genetic, we have to introduce the copy into the egg of a different animal. We have a species which lays eggs, like reptiles or chickens and stuff like that. And we need to introduce that human gene into the DNA or into the genetic material of the as well as the egg. Oh, oh, I don't know, man. I don't know what I was thinking. If I could study, if I could do something. But no! Oh no, it's wrong. Babe, these beautiful couches that you bought, you can't sit properly on them. I don't know what you were thinking. Why did we get these couches? And these ridiculous pillows. This interesting color scheme. Blue, brown, and this like weird beige thing. Oh god. It is 4:18. Yeah! Mm. That woke me up. That woke me up. That woke me up. Ahidaha. JavaScript practices are a funny bird game that harkens back to the Bible. Harkens mentioned. I'm so tired. I need to do this. Need to put this in the background of the music that I'm listening. It sounds like it's amazing. Oh, so good. Ah, shit. Alright, that's enough of that. Oh my god. Let's move. Yeah, I'm gonna go crazy considering. My fault. Okay, deep breaths. Oh. Oh. Just like that. Alright, the production of proteins using transgenic animals involves introducing a copy of the human gene which codes for the desired protein into the genetic material of an egg of a different animal. As well as the gene for the specific protein, there's also a promoter sequence which makes sure that the gene is expressed in the mammary gland of the female. The fertilized egg. What? 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 Okay, fine, right? Because the okay, fertilized egg now developing a transgenic embryo is replaced inside. Oh, egg cell. Okay, never mind. Genetic material of an egg cell of a different animal species, as well as the gene for the specific. There's also a protein sequence. Promoter sequence which makes sure that the gene is expressed in the mammary gland of the female. This is important. Fertilized egg now developing a shut up. Fertilized egg now developing a transgenic embryo. It's then replaced itself with surrogate substitute mother. Is born and grows to maturity. When the animal is mature and produces milk, the milk is harvested, purified. Fertilized egg now developing transgenic embryo is placed into a surrogate substitute mother. Because remember, this egg, the fertilized egg, was from a different animal. So when it's placed inside the surrogate substitute mother, it's born and grows to maturity. And when the animal is mature and produces milk, then the milk will be harvested and purified and the human protein extracted. So it's going to be growing from the milk because we said that the pharmaceuticals would make sure that the gene will be expressed in the mammary gland, all places. So far, scientists have succeeded in producing transgenic sheep, cows, pigs, rabbits, mice, all capable of producing human proteins in their milk. When we use large animals such as cows and sheep, large volumes of milk and therefore human proteins can be achieved, and the number of animals can be increased by the air quality. Examples of drugs from transgenic animals. Transgenic animals have so far produced more than 20 different human proteins, and some of them are already in therapeutic use or being trialed. Factor 7 and factor 9, I think, are both important components. Is that 9? Yeah. Are both important components of the human blood clotting cascade, which can be missing in hemophilia and other blood clotting diseases. These factors are being harvested from transgenic. Alpha-1 antitrypsin is the protein produced by tracing. Okay. The milk of such transgenic sheep contains up to 35 grams of different protein every liter, with a very, very high yield. However, the milk is very expensive. It costs several $1,000s per liter. Trials are in progress on treatment using the transgenic protein, and the idea is that the numbers of transgenic animals, as the number of transgenic animals increase, the price will eventually decrease. However, developing these products commercially has presented a problem. Most of the transgenic sheep were destroyed when the company that produced them met financial difficulties. What? Are you stupid? This Ridgeman? Okay. Lamayo? Just kill all the sheep. Activated protein C for treating deep vein thrombosis is also being tested. As we begin to understand the mysteries of the human genome in increasing detail, we should be able to identify the genetic sequence for every chemical. Oh, that's so cool. We should be able to identify the genetic sequence for every chemical in our body. If genetically modified organisms can make many of the healthy molecules for us, if genetically modified organisms can make many of the healthy molecules for us, whether they are bacteria, plants, or animals, the impact on future health and medicine can be enormous. Why do they use the word health? Okay, this one's only 2 pages. Or it's like 1 page. Okay. So let's finish this one and then we can take a break. The break before doesn't count because I didn't do anything in that break. Yeah, I did jack shit in that break. I wanna like well, it's gonna turn 5 o'clock in 20 minutes. That's when they give it on. I don't know what do you wanna do? Like we could crochet while watching EVG. We could crochet in general. I've done things and I mean I do need to coding. I do need to make a video for today. Even though I did make a video. I want to make another video. What if I use Discord as the thing? Like, what if I wait. What would happen if I use Discord as the thing to make journals? How would that? I know I was supposed to study first, but like, it'll take a second. Um, um, how do I do this without like doxing my Discord? Um, I don't know. Actually. Do I really care? Um, maybe a little. Let's see how it works. Okay, so no, that's not. What is it that Alt? Is it Alt which removes it? It does, but it also makes it bigger. Interesting. Okay. Well, okay. Okay. Kind of. Okay. How would the text look if I wrote something down there? Boom! Kaboom! Hello. Can I increase the size? This is an attempt to use something besides Notepad to make the. Hey, it works! It's not bad. It's kind of like a fun little idea now. Oh, something like this maybe? Let's say Hey, I like it. Maybe actually. Okay, maybe this could work. Okay, cool. It's like if I didn't want to write too much in one message or something like that, maybe it could work as a more casual format for the video, as it uses a kind of texting public rather than a big journal or digital journal. But what would you be reading? Would it be what I'm typing down here, or what's gonna pop up on the actual, like, you know? Hmm. Cool idea either way. I like it. It's pretty cool. Anyways, let's get back into this. Okay, so microarrays and bioinformatics. Know what microarrays are. Know how microarrays can be used to identify active, active genes, like ones which are being like transcripted more of the time. Understand what is the term, what is meant by the term bioinformatics. Scientists need to be able to identify the individual for many reasons, including genetic modifications, gene sequencing, identifying gene mutations which cause diseases, or analyzing patterns, genes, and diseases within a population. Scientists have developed one very useful laboratory tool which allows them to detect thousands of active genes at the same time in the microarray. Okay. When genes are active, they are expressed. The mRNA is produced from the active genes and is used as a template for production of amino acid chains in the cells of ribosomes. If there is a mutation in the gene, a different form of mRNA will be produced. However, okay, if there's a mutation in the gene, a different mRNA form will be produced. However, if the gene contains a very large amount of DNA bases, and there are many places where a mutation, and there are many places where a mutation occurs. Microarray is a tool which scientists use widely to show if a DNA sample from an individual contains any mutations. These samples are also known as DNA checks. Okay, so we have a DNA chip and we use a microarray to see if that DNA chip has any mutations. Basically, a DNA microarray is a slide on which there are thousands of spots. Each spot is in a specific place and contains a known DNA sequence. The mRNA sample are then collected. Scientists usually use a reference sample with a known gene sequence and an experimental sample. Typically, this will be from an individual with a particular disease which may have a genetic element, for example, a form of cancer. Okay, so then I guess they just try to find the difference between the two samples using a microarray. Reverse transcriptase enzymes usually convert the mRNA to cDNA. Each sample is given a first label. Usually, the known sample is given a green and the experimental is given a red. Labeled DNA sample are mixed together and applied to microarray slide, where they bind to the matching DNA probes. This is called hybridization. After hybridization, the microarray is formed. How do people make terms like this, and why did they call it hybridization? Anyways, after hybridization, the microarray is scanned to measure the fluorescent light which produced in different spots. If both samples are expressing a gene equally, the light will appear yellow, which is a mixture of red and green. If the experimental is expressing more than the control, if the experimental is more than the control, then it will appear red. If the sample is expressing less than the control, then it will appear green. We can collect very large amounts of data using technology. Analysis of the data provides full information about key profiles. Okay, so it's profile. Right, this is the is it or the sequence point? The analysis is detailed information about the profiles, the causes of many diseases, and the effectiveness of some treatment. Microarrays in breast cancer. Can you see? No one did this? Oh, bro. Let's open up. Mm. Oh wow, they are super small. Okay. Mm. Okay. Some forms of breast cancer respond very well to treatment with drugs which block estrogen receptors or prevent estrogen synthesis. Doctors can use microarrays to show the level of expression of the gene for estrogen receptors in individual parents. If it is high, then the patient will respond well to these drugs. Breast cancer, they respond very well. Some forms of breast cancer respond very well to treatment with drugs which block estrogen receptors or prevent estrogen synthesis. But when we use drugs which do these two things, block estrogen and receptors and prevent estrogen synthesis, then um, the breast cancer can be defeated, or it can be treated. Now, when we use microarrays to show the level of expression of the gene for estrogen receptors in the individual parent, if it is high, the level of expression, then the parent will respond well to these genes, and they will increase the chances of responding. If the patient shows low levels of expression of the gene of estrogen receptors, then the estrogen blocking drugs will not be effective and are not given. Why? Right? Because if we have high expression, then we know that those are being expressed a lot, and so we need to give drugs which block them. But if they're already low, then why would we give drugs which block everything if the expression of those genes is already very low? Now, in this case, different approaches to the treatment must be used. So we have cell samples, but this using a DNA microarray. We have a normal cell and we have a cancer. We make cell cultures from the cell samples. Take the mRNA isolated from, we isolate the mRNA from these cells. We give we use reverse transcriptase and these for labeling, like I guess it's like gene markers or whatever. And we make the mRNA from the normal cells green, and we make the mRNA from the cancer cells red. And then uh, the DNA probes hybridize this with all these probes. Hybridize these to align sequences on my chart. Got it. And we see not present on in both types of cell. And this is a normal cell for me. Like this kind of sequence is a normal cell for me. This kind of sequence is in non-building in disease cells. These two are the ones that we should be looking on. These two is where the genes differ. See if I actually got that. Oh, 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 oh. Okay. It almost a devil. Yeah. Label DNA samples are mixed together and applied to macros where the find to match the main plot. What does this all tell you about how does the result how does the result tell you about whether or not a gene is highly expressed? If it's present in healthy but not disease, therefore in disease it is expressed less. Let's break down the storm using a bit of complementary base theory and logic. We're taking a shower now? Have you played Esther? This has been a long time ago. It's been Esther. I'm gonna go to the monster bed, but we should probably pray it first. If you want to turn the heater on, then what I recommend is you like turn on the heater, you wudu pray, and then you go take a shower. It's fun what I do. You don't know what I'm trying to tell you. Anyways, if I miss this, bro, I'm gonna kill myself. Where does go? When did they get gone? I could just go early. Okay, let's just finish this then. No, molecular velcro. A DNA microarray is a slide with thousands of tiny spots. Each spot contains many identical single-stranded DNA sequences called probe. Each spot contains many single-stranded DNA sequences called probes. Each probe contains a specific represents a specific gene. This is a process where a single-stranded cDNA sample is the one you may find and bind to the complementary probes onto the slide. Because DNA naturally wants to be double-stranded, the cDNA sticks to the probe that matches its sequence. The diagrams you provided, the normal cDNA is labeled green and the cancer is labeled red. So interpreting the color code. So basically the microarray it contains an mRNA strand, and each little probe thingy contains a gene of that strand. And so the cDNA over here, which is complementary to like each gene which is complementary to each gene here will bind. Each gene here that is complementary with each gene here will bind. So over here, this one and this one, or rather this one and this one, you can see that the red ones contain like some sort of base pairing sequence, which is complementary to the base pairing that are here already. Okay, but for this one, none of the genes that were present here were the black ones. They were not present in any other of the other ones. They weren't complementary to any of the ones over here, and so that's why it's not present over there. So then the intensity and the color read spot tell you the relative expression of a specific gene. As an example, since cDNA is based on mRNA, the more mRNA itself produces for a specific gene, the more cDNA will be available to stick to that spot. I see. So only the normal cell cDNA is hybridized here. The gene is expressed in fully cells with silenced or downregulated in disease cells. Okay. Since the green spot is present only here but not in the red spots, this means that it is silenced in the disease DNA. This gene. Anything which is green and not red, that is going to be silenced. And everything that is red which is not green is going to be expressed, but it's going to be activated. So highly expressed upregulated in the cancer cells, not the healthy ones. Both samples mentioned the gene is expressed at the same level. So this gene is not affected. And no cDNA hybridized, this gene is not expressed in either cell type. So again, nothing changes. Thus, present in healthy but not diseased mean less expression. Precisely, if the spot is green, confirms that mRNA. A red spot indicates an oncogene, a gene that promotes cancer when overreactive, while a green spot represents a tumor suppressor gene. The cancer cell is successfully switched off to allow uncontrolled growth. Bioinformatics, DNA technologies such as microarrays, DNA sequencing, and DNA profiling. Okay, we're bringing them all together, okay? Microarrays, DNA sequencing, DNA profiling, these we learned last year. Generate huge amounts of data, far more than was ever produced in the past during scientific investigations. Individual scientists or even teams of scientists could not possibly analyze these to search for patterns. This is why the new science of bioinformatics is important. Bioinformatics is the development of software and computing tools needed to organize and analyze raw biological data. This includes the development of algorithms, mathematical models, and statistical tests which can help us interpret the enormous quantities of data that are generated. Using bioinformatics, we can process and use the information generated using microarrays and other forms of DNA analysis. Bioinformatics enables scientists and doctors to use the information from these DNA techniques to adapt the care of individual. Bioinformatics enables information from the DNA to adapt the care of individual patients and learn more about biology. This is low-key kind of my thing. This might be what I'm gonna do. This might be what I'm gonna do. Bioinformatics. This literally might be my thing. Bioinformatics. How did you get into bioinformatics and how happy are you with it? I'm a freshman looking for fields to specialize in, and bioinformatics interests me as someone that has an interest in computer science and biology. but I'm worried it's the wrong field and I'll be stuck without a job or unsatisfied with my career. So I just want to ask more broadly, what led you to this career path, and do you regret anything about the path that you've taken? Any responses would be greatly appreciated. I'm a software engineer and want to pursue bioinformatics because my mom passed away by cancer. I want to fight my best battle. With a PhD in bioinformatics and now working as a research engineer, works well for my personal and career motives. I now architect and develop population scale health and genomic solutions for the fun stuff. My background is computer science and engineering. My PhD is computer science algorithms for metagenomics. That is so dope. Yeah. So most people in any field don't have perfectly linear trajectory into their career. If you focus on your interests, values, and develop the transferable skills, you can pivot into new fields and opportunities as they present themselves. Much like I did with bioinformatics, I started in biology and did some graduate work that required bioinformatics. I didn't work I didn't work as a bioinformatician until staying in academia for 5 plus years. I switched during the pandemic because work aligned with my values and interests. I wanted to WAFH work from home and get paid more, among other things. If you focus on developing transferable skills, you'll never be stuck in any field slash career. Okay, I actually would love to learn some transferable skills. Is bioinformatics still worth pursuing? I'm currently pursuing a bachelor's degree in bioinformatics in Barcelona. After reading a lot about discussions in the job market, I've noticed a lot of people saying that a PhD is often required in this field, and even then, many graduates end up staying in academia due to limited industry opportunities. I'm worried what alternative career. Okay, what is academia? Isn't academia like research? Academia, the scientific and cultural community engaged in higher education and research taken as a whole. Um, so is being stuck in academia a bad thing? I'm worried. I'm wondering what alternate career paths I could pivot toward that are related to bioinformatics but may offer stronger industry prospects such as biotech, pharma, software engineering, or other adjacent fields. Has anyone made any similar transition or have some skills or areas I should start focusing on? I'll be direct. A bachelor's in bioinformatics on school is weak leverage in Europe right now. Industry roles are limited, geographically concentrated, and often prefer either PhDs with deep biological expertise or candidates who are fundamentally strong software engineers or statisticians. A general bioinformatics degree can end up too shallow in biology for research roles and too shallow in computer science or math for competitive engineering positions. That's a structural trap. PhD required narrative the PhD required narrative isn't entirely wrong either. Many genomic and computational biology roles in pharma strongly prefer or require one, and industry absorption is narrower than people expect. The key isn't the degree title, it's your skill set. If you want strong look at the job scenario, see what the requirements are, and take note of how many are truly entry level. In general region, almost every analyst job says master's required PhD preferred. A lot of people haven't applied to jobs since 2024 when things went downhill. They have a totally different view of things because they work with people who have master's degrees, got hired 5 years ago. Companies don't want to train people in all sectors. Just an idea of how it is in your area and go from there. I I had applied to bioinformatics and data science jobs. Anecdotally, I just started applying myself. I've gone to all Python and data science, ML engineer jobs, and none for bioinformatics jobs. Are there? I got my PhD in bioinformatics, which is very difficult due to lack of funds. After completing it, I didn't want to speak of this to my PI ever again when I went to industry. Now in industry, it is a big plus in the PhD in the company I work, even today as a bioinformatician. But the other companies didn't value it at all. Fact of max one of the companies can value your PhD. The final line is I have a bioinformatics position and I basically have become a software engineer. All pipelines need to be something nowadays, and therefore the companies just buy them as software from other ones. So PhD is a pain in the ass, and we could not do it again. You could work in another informatics field, like what? Geoinformatics. Comments plus one plus one. Barcelona right now is another good place to be. Actually, it's a bioinformatics hub for Spain's companies and includes the supercomputers in Europe. That being said, as an entry level, you'll most likely have an extremely hard time finding a job after an internship.
or expand your experience in a related branch before giving full comeback to bioinformatics. What related field that is really up to your preferences and talents? Any of those you mentioned are viable. I don't think a PhD is required for this field. In fact, it's one of the few technical fields that don't necessarily care about one, from what I've seen. Academia might be looking for a computational postdoc, but aside from that, I see plenty of places looking for analyst roles, which is the general term. You can work in a lab specifically doing bioinformatics, or a core RB bioinformatician for a non-dry lab. National labs and plenty of companies also hire a lot of analysts. Point is, you'll want to do your diligence to gauge what kind of work you'll be doing. Above all, having experience in a visible portfolio is going to make it competitive, whether it be publications, manuscript writing, or juicy GitHub. If you strictly want to know alternative roles that aren't specifically biology but have many potential skills from bioinformatics, hmm, biology but have data science roles, which these are, and machine learning DevOps job roles, except for anyone working with data. Consider it something by most people that will complain about the job market are people that struggle to find work. It could be for a variety of reasons. There's so many posts that ask if it's worth pursuing. Sometimes I urge you to not pursue something, not only if it's worth it, but pursue something if you actually want to. I don't know, because every job I look for competition by informatics is too far to be. The market is already tough and saying job. Obviously, a PhD doesn't hurt, but it's not needed by any means. Maybe it's depending on the country, but in the US and how many positions that last PhD. Yes, with AI, the possibilities are endless. The problem is you can bring solutions to the optimization. This is where the innovation starts. Cures, medicine, feeds, food. Mm, But the hard ones for the hard ones. Personally, I think it makes much more sense to go all in tech to cast for one and then drill down into the. Yeah, yeah. Okay, good read. Wow. Bioinformatics career r/bioinformatics career. Good read, good read. Okay, I'll be back. Actually, no, I won't be back. Yeah, no, the bioinformatics. Yeah. Okay. Yep. There you go. Have a nice day.